All right, so the second part of this chapter now we're going to talk about what is the mechanism by which mRNA is translated into a polypeptide. So the secret to this, in order to know this, you have to know about ribosomes. So we've talked about polymerases, so DNA polymerases copy DNA, RNA polymerases turn DNA into RNA, and so now we have the ribosome. And so the ribosome is what reads the mRNA, and it translates that, and it creates the polypeptide from that. So these ribosomes are very, very large structures, so you can see them under electron microscopy pretty easily, and they consist of a large number of proteins, so these are unique proteins, and also RNAs as well. So depending on the kingdom of life, there are either 3 or 4 rRNA molecules within this, and then there's between 40 to 80 ribosomal proteins. So there's a huge number of proteins that come together to assemble this, this large superstructure. So the ribosomes bind to the mRNA, they create the polypeptide chain, and this ribosome is broken down into 2 pieces, a large and a small subunit. So we'll talk about that more in a second. And then you also need to know about transfer RNAs. So we talked about tRNAs. So tRNAs are very small RNAs. They're about 80 base pairs long, but they are what actually are responsible for creating the genetic code. So we know the genetic code, we solve the genetic code, we know if I gave you an AAA, you'd be able to tell me the amino acid. But something within the cell has to enforce that, and that's where the tRNAs come from. So they actually have 2 very specific parts. One part recognizes a codon, and then one part binds to a specific amino acid. And so that's how that genetic code is enforced. So first, we'll talk about the basics of the ribosome structure. So we'll distinguish again between prokaryotes and eukaryotes. And so the monosome is a large structure called 70S in prokaryotes, 80S in eukaryotes. And they look very similar to each other. So in both cases, you have the smaller region right here, the smaller region right here, and then you have 3 gaps within it. So there's 1, 2, 3 gaps within the ribosome, 1, 2, 3 gaps within it. So we break this apart into 2 pieces because they can actually separate there. So we have the large subunit, which is the bottom, the 50S, and then the small subunit, that's the 30S. And then in eukaryotes, it's the 60S and the 40S, mainly because they're a little bit larger. The eukaryotes have more proteins and longer rRNAs. And so you can just see like how many proteins are actually used to create these large subunits. So there's a crazy number of proteins, 31. There's the 23S RNA, the 5S RNA, and then the small subunit has the 16S and then 21 other proteins. So the reason that there's either 3 or 4 rRNAs is that in eukaryotes there's 2 smaller size rRNAs, the 5S and the 5.8S. So one thing that might be useful to remember is that the small subunit has the middle size rRNA, the small subunit has the middle size rRNA, and then the large subunit has the large and the 2 smaller, or the large and the smaller. So the tRNAs, we've seen these before. These are small in size and very stable. They're about 75 to 90 nucleotides. We talked about before, they're transcribed from DNA, they're processed. They are often transcriptionally modified, post-transcriptionally modified. So many of the nucleotides have unique modifications to them so that they're not just an A, C, G, or a U. And all of these all of these tRNAs, and there's a large number of them, all of these have a cloverleaf structure. So they have an acceptor arm, they have a amino acid binding site, and this is at the 3' end. They have this arm, the TC arm, and they have the D arm, and then importantly they have the anticodon arm. And so the genetic code is determined by matching this set of 3 nucleotides, which are responsible for binding to the mRNA, with this part right here, which is covalently bound to a specific amino acid. So what do we mean? So here are an example of two different tRNA types. So one is tRNA phenylalanine, one is tRNA proline. And so, these are encoded separately on the genome. So they have different sequences to each other. They're different genes. But one of these has a AAG at the anticodon arm, and one has a GGC at the anticodon arm. And so these are genetically encoded on the DNA, these differences, and these just are these three particular nucleotides right here. So this tRNA is able to recognize the UUC in the phenylalanine. So the 5' is here, the 3' is here. And so we have anti-parallel bonding, and then you'll notice that it's complementary. So the G binds to the C, the A binds to the U, and the A binds to the U. And so this tRNA, this particular tRNA, is able to recognize and bind to a codon in the mRNA that is complementary. This particular tRNA is always attached to an amino acid, a phenylalanine amino acid. This tRNA has a different anticodon, so it is a CGG, if we read it in the 5' to 3' direction, and so it's able to recognize a CCG codon in the mRNA. And because this tRNA is always attached to a proline, then whenever it is used, it always adds a proline to the growing chain. So this is what we mean when we talk about the mechanism of the genetic code is determined by what three nucleotides the tRNA has at the anticodon position and what amino acid is always bound to the end of it at the 3' end. So tRNAs are named according to the amino acid they bear. So tRNA phenylalanine, tRNA proline, and then again the anticodon is anti-parallel to the codon. So tRNAs share many features. So they all have these different stem loops. They all have these anticodon region, and they often share variable sites. So you can see that there's a bunch of different types of modified bases. The methyl inosine, the inosine, etc. So they kind of all have these like little designations in the same way as A, C, G, and T refer to distinct nucleotides, ribonucleotides. These refer to specific modified ribonucleotides. And you can see this shows you the actual chemical structure of these. I don't expect you to know the chemical structure of any of these modified bases, but you should be aware that there's extensive modification kind of all over it. So we have a couple modified bases here, we have a couple modified bases here, etc. And so there's also modified bases that occur at the anticodon loop, and importantly, you can also have a modified base as part of the anticodon site. And this becomes important for the wobble hypothesis, which we've covered before. So charging tRNA. So I said before that each of these specific tRNAs is attached to a specific amino acid. And so how does that happen? So this is not something that occurs during transcription. You can transcribe a tRNA molecule, but an amino acid is not a nucleotide. An amino acid is a completely different type of molecule. So somehow you have to know, somehow this tRNA has to know that I can only attach to a phenylalanine. And so this process is called charging the tRNA. So amino acylation is tRNA charging. And so before translation can proceed, the tRNA molecule must be chemically linked to the respective amino acid at that 3' end. And so these enzymes right here are very important for that process. And so these are amino acyl tRNA synthetases. And so these are the enzymes that recognize particular tRNAs, recognize a particular amino acid, and then attach those together. And so there's 20 different synthetases, one for each amino acid. So proline has one, phenylalanine has one, etc. And so each one of these So let's see. You know, you have this tRNA, and then you have this synthetase come by. And so this synthetase is specific to phenylalanine. And so it's able to bind a phenylalanine amino acid. This synthetase also recognizes the correct tRNA that has the AAG, and then it is able to attach the phenylalanine to the tRNA. And then you would have a completely unique molecule for the tryptophan amino acid, completely different synthetase for the alanine, et cetera, et cetera. So each one of these is very specific, recognizes a single amino acid, and then recognizes the tRNA that it needs to charge. So charging it, so the activated enzyme complex takes the amino acid, and it also takes ATP, which is used for energy. And so initially you have a reaction that activates the complex so that the amino acid is bound to it. It also has the ATP to use as a energy source, and then it's able to use that energy source, cleave the phosphates from it, and then so it cleaves the phosphates here in order to create an energetic modified version of the amino acid, which it can then recognize the tRNA of interest and then add the amino acid to that. So that's the process of charging a tRNA. So there's a specific enzyme that's required for charging tRNA. So there's there's multiple tRNAs, there's multiple synthetases, and then they act together in order to create a fully formed charged tRNA. So once you have a fully formed charged tRNA, then you are ready to start with initiation of translation and progression of translation. So you need a ribosome, you need certain initiation factors, you need elongation factors, you need release factors. So this is similar to transcription, where there's initiation, elongation, and termination. And there are specific factors that are involved in each. So you need tRNAs, and then you need an mRNA in order to do that. So when we start with the mRNA, the first thing that we need to have happen is we need to have a initiation to occur. And so initiation requires a very specific tRNA called the initiator tRNA. And so the initiation codon is an AUG, ATG on the DNA sequence, and an AUG in the AUG in the RNA sequence. So this mRNA has to have an AUG that indicates the start. And so this anticodon must bind in a anti-parallel direction with the AUG. And again, this tRNA has this modified methionine. So the first, at least in bacteria, the first amino acid that gets added to a polypeptide is always this modified methionine amino acid. And so for initiation to occur, you have to have these initiation factors, and you start out with the small cap. These initiation factors bind the mRNA. They help the cap bind the mRNA, and they also help the initiator tRNA initially find that proper codon in the mRNA. So you see we have this anti-parallel bonding between the AUG and the UAC within the tRNA. So once this forms, now we can have the complete formation of the ribosome. So some of the initiation factors release, and then the large component of the ribosome can also occur. And so now we're ready for elongation to begin. So in bacteria, ribosomes recognize the Shine-Dalgarno sequence. So the Shine-Dalgarno sequence precedes the AUG. So again, we talked about this being in the 5' UTR. And then there's base pairs within the region of the 16 RNS that facilitate this initiation. So the Shine-Dalgarno sequence is before that in the mRNA, and then it is able to help initiate the formation. So what happens during elongation? So now we have to have this process where the mRNA kind of moves through the ribosome. So this mRNA has to move all the way through the ribosome, with each step incorporating a new tRNA and a new amino acid into the polypeptide chain. So we've started with the initiator. So the initiation occurs within the P site. So remember, we saw that there was these 3 channels. So the P site is where the polypeptide occurs. The A site is the active site. So this is where the new tRNA enters. So this is the tRNA that binds to the codon just to the 3' of the previous one. And so now once it has entered, now we can covalently bond the amino acids that are both on the tRNA next to each other. And so kind of simultaneous to this, the tRNAs move direction. The mRNA translates through the ribosome. This pushes the tRNAs over as well. And so now one of the tRNAs enters the E site. So this is called the E site. And the other one that was formerly in the A, the active site, now moves to the P site, the polypeptide site. And so this contains the polypeptide. So this tRNA has lost its amino acid, and this one now has 2 amino acids. So now it's moved through, and we're ready to incorporate the new tRNA into it. And so this tRNA enters the active site, and then we're ready to now add this amino acid that's attached to here to the polypeptide chain. So again, that happens. So it's actually this polypeptide chain that gets moved to the tRNA into the active site. We move through, so this tRNA then enters the exit site. What was in the A site now enters the P site and has the polypeptide chain, and we do this over and over and over again. So as elongation occurs over and over and over again, we keep getting a longer polypeptide, and the most recent tRNA to have entered the ribosome is the one that actually holds on to that entire polypeptide chain. And we keep moving through, so the mRNA keeps moving through the ribosome. So finally, we reach the termination phase. So the termination phase happens when you have a UGA within the mRNA sequence. And so at that point, the termination codon is in the active site because the mRNA has moved through. And so now there is no tRNA that recognizes this codon. And so this allows certain release factors to stimulate hydrolysis and then release of the polypeptide chain. So RF1 and RF2 are the 2 factors that are responsible for the termination of translation. And so now we have release of this long polypeptide chain. The primary sequence, we know which was determined by the mRNA, and now that polypeptide chain is now ready to start to fold, form the alpha chains and the beta sheets, and then form its final tertiary structure, and then potentially interact with other proteins as well to form quaternary structures as well. So the tRNA, study of the tRNAs also provided us with a molecular explanation for the wobble hypothesis. So why is that? So the wobble hypothesis says that the binding between this G, which is the 5' in the anticodon in the tRNA, with the 3' nucleotide in the mRNA, it says that this binding is kind of wobbly. The idea is that it's kind of wobbly. And so, you have this nice bond that's formed in between these sets of nucleotides, but this third one is kind of loose. And so this allows some sort of flexibility. And so There's also modifications that often occur of this anticodon base, and that also makes it easier for this tRNA to recognize multiple codons within the mRNA. And so, you know, what this table right here shows you is, you know, these are all of the nucleotides that can recognize a particular amino acid. So the G, you'll notice that if there's a G in the anticodon, it can recognize the C in the mRNA. But it can also recognize the U in the mRNA. So the G has two different mRNAs that it can actually recognize. The C's, you'll notice, can only recognize the G, but there's also ones like the I. So the I can recognize three different ones. The U's can recognize multiple ones. And so this provides a molecular explanation to the degeneracy of the code. So the reason that there are multiple amino acids, or there are multiple codons that encode an amino acid, is because of this wobbliness between the tRNA and the mRNA, particularly at that position. So this is the extent that we will cover the translation, and then the final part of the chapter will go over the one gene, one enzyme hypothesis.